And in a moment, I'm going to ask Dr. Benjamin Ebert to come up and begin with his talk entitled The Origins and Evolution of MDS. Dr. Ebert has made some critical, important contributions to our understanding of MDS and, in fact, has branched out beyond the hematology aspects to understand the role of inflammation and try to understand some of the cardiovascular events that occur in patients with clonal hematopoiesis rather than talk incessantly about his contributions in career to date. I'm going to invite him to come up. He is at the Dana-Farber Cancer Institute, where he leads the either called the Hematology Oncology, I'll just call it Hematology Oncology program. Ben.
Thank you, Steve, for that introduction, and thank you very much to the organizers for the invitation to speak here. This is really, has always been one of my favorite meetings. I think my first one was in the Edinburgh meeting, and that too, I think, had dancing. There was some Scottish dancing following the whiskey tasting, and images of some of my colleagues dancing there are permanently fixed in my brain. Hopefully, there'll be similar memories formed on Friday night. But this has been a place where many important collaborations were started, many friendships made, and I look forward each time coming back here to see old friends. So I'm looking forward to this meeting as well. So my disclosures are not relevant to this talk, but I've worked with Grail and Celgene and Deerfield, but not on the topics that I'm going to talk about. So just as a very high level, one slide that's now a few years old of what the genetics of MDS look like. Our IDWGPM molecular group will have a much more sophisticated view of this. But just to say that at a high level, the number of genes that are mutated in MDS are a bunch, but finite, not an enormous number of genes. Hard to learn every single one in their biology, but not an infinite number. And that they are co-mutated. In general, most MDS cases are not caused by a mutation in a single gene, but by a combination of mutations in multiple genes, and that those combinations of mutations happen in non-random ways. There are genes that are co-mutated more commonly than expected by chance, and some that are mutually exclusive. And the genotype matters. It's certainly what drives the pathogenesis of the disease, and the particular genes that are mutated predict the hematologic phenotype and an outcome, and in some cases response to therapy, which is the basis for many groups studying them. So it's important to note also that it's not all of what drives the disease. Different cells can have different epigenetic states. The mutations may have different cells of origin, which may be reflected in their epigenetic state. The bone marrow microenvironment may be different in different individuals, and that may influence the course of disease. The number and status of the normal hematopoietic stem cells may have a big impact on the phenotype of the disease. Many of those features are read out in our standard pathology. Things like cytopenias and morphology may reflect some of those abnormalities. We don't have ways to assess, for example, normal hematopoiesis or the microenvironment yet, but these are, in aggregate, I think, the primary drivers of the disease. Now, since MDS is caused in general by multiple mutations, these mutations may occur if these columns are hematopoietic stem cells. There might be one that gets an individual cell that gains a somatic mutation that leads to a clonal expansion. And only one of those cells will then acquire the second mutation, lead to a clonal expansion, and then additional mutations are required on the way to developing full-blown MDS and, in some cases, secondary AML. And those, one sort of conclusion that you might draw from a diagram like this, for one, all the mutations don't occur at the same time and in one fell swoop. And there, in general, is going to be a large clonal expansion of that first mutation, providing a larger number of cells that are available for the subsequent mutations to occur. And since the rate of those subsequent mutations may not be all that high, there may be a much, much larger number of individuals with initial clonal mutations than there are individuals who ultimately develop myelodysplastic syndrome. And our field in general has been able to study these premalignant states more readily than other tumor types because of the availability of sequencing data from peripheral blood. Our first foray into this used exome sequencing data that was acquired at the Broad Institute in about 17,000 individuals for completely unrelated reasons. They were looking at the genetics of diabetes and cardiovascular disease and other disorders, but were committed to openness of data and sharing, and the people who created those data hadn't occurred to them to look for somatic mutations. They were interested in germline predisposition to disease. And so we took those data and looked for somatic mutations, specifically looking for mutations in genes that we know to be drivers of hematologic malignancy, and found evidence of these mutations even in the exome sequencing data that was extremely age-dependent, and individuals under 40 very, very rarely had mutations in their peripheral blood, but by the age of 70, more than 10% of everybody had a mutation in their peripheral blood in one of the genes that leads to MDS. It turns out that these mutations that we found were really the ones that we find in MDS and in AML, not the mutations that we find in lymphoid malignancies. So this is a very common state. Not surprisingly, those with a mutation in their blood had a higher probability of developing a hematologic malignancy. If we think about that as the very first step on the way to getting a full-blown malignancy, it's not surprising that having a first hit increases the likelihood of developing a full-blown hematologic malignancy, including MDS or AML. A very exciting part of this field has been the convergence of data from many different groups that has been extraordinarily concordant. So these are a couple graphs from a paper from Steve McCarroll's lab, who was actually interested in the genetics of schizophrenia and stumbled across the same finding where the same genes were mutated at same types of frequencies, same age dependence, same association with hematologic malignancy. Now many of, and right in that very first wave, papers from WashU and the Sanger Center in Cambridge, and then subsequently many, many other papers. Easily more than 100,000 exomes have been analyzed, or samples have been analyzed now, and so we have a really deep understanding, at least of the genetics of clonal hematopoiesis, and work on the biology and clinical consequences is ongoing very rapidly. Wanted to say a couple things about kind of the different types of clonal hematopoiesis. In the red bars there are the number of individuals with somatic mutations in the drivers of myeloid malignancies. This is from a paper from Decode Genetics. And in purple are the ones where using whole genome sequencing, they did whole genome sequencing on 10,000 individuals and looked for clonal hematopoiesis in those. They could see clusters of somatic mutations that are passenger mutations, for all we know, that are all around the same allele fraction and provide very robust statistical evidence that there is a somatic clone of particular size in those individuals. And that's far more common than the number of individuals who have known driver genes. So they have clonal hematopoiesis, not the kind that we can detect readily in the clinic, short of whole genome sequencing, but certainly have clonal hematopoiesis. They have evidence of a clonal process. In addition, studies from WashU and elsewhere have indicated that if you sequence deeply enough, you can start to find TET2 and DNMT3A mutations in essentially everybody with age. So ultimately, clonal hematopoiesis could be considered present in all of us and everybody, and then it loses any meaning. So we need to define some limits of what we're thinking about, at least on a clinical level, on a scientific level. We're interested in all these types of clonal hematopoiesis and what they might mean. But it does mean that many, many individuals, the vast majority of individuals, will develop a somatic mutation, even with one of the drivers, say DNMT3A, and it will sit there at a tiny clone size, potentially for years or decades. So we took a first stab at defining some features, and I'm sure that this will be revised repeatedly over the years, but wanted to make sure that this is distinct from a known diagnostic process, so that there's no morphologic evidence of MDS in particular, and that they don't have other established premalignant states like paroxysmal nocturnal hemoglobinuria, MGUS, the precursor to myeloma, or MBL, the precursor to CLL. And we thought we needed to set a threshold for the allele fraction. We set it at 2%, though that very much could be adjusted. So had to have a mutation in one of the drivers of myeloid malignancies present in an allele fraction of at least 2%. In fact, many of the clinical consequences, including development of a myeloid malignancy, are greatest in individuals with even larger clones, but at least that's a starting point and easily within the range of detection for most clinical assays. I don't think it's unusual to have a situation in medicine where we need to draw an arbitrary threshold. Hypertension, high cholesterol, depression, diabetes, almost all of our diagnostic categories in medicine are on a continuum, and we need to draw some boundary between normal and pathologic that is an artificial boundary, but we will need to draw one to some degree here so that we can all talk about the same thing. And with those types of limitations, the odds of progression from CHIP to an overt malignancy is about 0.5 to 1% per year, and even higher if you take the individuals who have larger clones. So this process, this concept of initial mutations leading to a clonal expansion in individuals who do not have cancer is not limited to the blood and is probably a common feature of the pathogenesis of cancer very broadly. This has been analyzed very elegantly in the skin this is from blepharoplasty samples. That's a little sliver under the eyelid that is sometimes removed for cosmetic reasons in older individuals, and doing many, many, many tiny biopsies and sequencing them, and then recapitulating that virtually in that diagram on the right of what all those clonal expansions are. So those are clonal expansions of cells with known driver mutations for skin cancer, not melanoma, that are very much like clonal hematopoiesis, with the exception that in solid organs, there's a spatial constraint. Those clones can't get infinitely bigger as hematopoietic stem cells can by circulating. This is clearly true in the esophagus. It's likely true in many organs, and I think starts to change a little bit how we think about the genesis of cancer, that we are full of small clonal expansions in all our organs, and cancer doesn't come from just a single site developing multiple mutations at the same time, but one of those clonal expansions then acquiring a subsequent mutation, then additional ones leading to a full-blown malignancy. And that has a lot of implications for how we think about early diagnosis of cancer. So the other exciting aspect for us of this field has been that there may be consequences of clonal mutations that are not limited to hematologic malignancies, and that is likely true in MDS as well, that there are consequences of somatic mutations in the blood that are not limited to the development of leukemia. The vast majority of patients with MDS, in fact, don't die of leukemia. So the first thing that we looked at, because we had the data to do so, was overall survival, and found that those with CHIP had about a 40% increased risk of dying of all causes, which is a fairly significant shift in all-cause mortality, and far greater than we could explain by the development of hematologic malignancies, which, despite all of our interest in MDS, is relatively rare and hard to shift overall mortality rates just on an increased risk of MDS or AML. And this is held up in many data sets and is a very robust finding. We looked for the cause of this. It turned out we did not see a signal for increased risk of solid tumor deaths, but we did see, much to our surprise, not something we were looking for at all, was that CHIP was associated with a higher risk of myocardial infarction or ischemic stroke. And the hazard ratio was fairly substantial, with a hazard ratio of about 2. As a frame of reference for these particular data sets, the hazard ratio for hypertension and hypercholesterolemia were about 1.4 to 1.6 in these. Diabetes was greater, but this is as great as many of the established risk factors which we were controlling for in this study. Still, This was, a retrospective analysis where we were fishing around our data to try to understand why we might see a shift in overall mortality, so subject to many concerns statistically. So we spent a while sequencing 4 additional large cohort studies to validate this finding. These are 2 prospective cohort studies that were developed in significant part for the understanding of cardiovascular disease. In fact, the second one, MDC, is the Malmo Diet and Cancer Study, so that's just a few miles over the bridge from here. We had blood samples from this is a prospective study, so we're looking at blood samples at the beginning of the study. Then these patients were followed for years. We knew who developed a heart attack and who didn't, and we could sequence people who did develop a heart attack and match controls. And our initial findings were replicated almost precisely with a hazard ratio of 1.9. We also looked at 2 cohorts of early onset MI, and the effect side was even greater. Clonal hematopoiesis is extremely rare in 40-year-olds, but in those 40-year-olds who do have it, the risk of myocardial infarction is much, much greater, particularly in those with JAK2 mutations. So that didn't prove that there's a causal association. We know that CHIP is very age-associated, and we know that cardiovascular disease is a defining disease of aging. So to demonstrate causality, we did this in a mouse model, and now many labs have done this with extremely concordant results. Mice don't get cardiovascular disease in normal wild-type mice eating a normal mouse diet, but if we knock out the LDL receptor and feed them a high-fat diet, they get very high cholesterol levels, and they do get atherosclerosis. And we can vary only the genotype of the blood cells to transplant in either wild-type cells or transplant in TET2 mutant cells and see whether TET2 loss just in the blood cells is sufficient to alter the development of atherosclerosis. And indeed, it does. So in the bottom here is the descending aorta of a mouse with wild-type controlled blood cells. Here is mice that have TET2 heterozygous knockout, and these red lesions are stained with oil red O and are the atherosclerotic plaques. That's staining the lipid-laden macrophages. And these are mice with homozygous TET2 knockout. The same findings are being made now with other genotypes. Same findings are being made with mice that have only 10% of the cells with the TET2 mutation, 90% wild type. So this has held up multiple laboratories, multiple genotypes, and variation in experimental protocol, but very reproducible. What might be causing this? This is a very, very active area of an investigation, but this is simply RNA sequencing from macrophages with loss of TET2, and there's a fair bit of literature that TET2 loss is relevant for the expression of inflammatory cytokines and an inflammatory state of those cells. But we found that when we did gene expression profiling, in particular after exposing cells to LDL cholesterol, we saw increased expression of a number of inflammatory cytokines. One particularly interesting cytokine is interleukin-1 beta, and that cytokine is interesting because there's a drug that targets interleukin-1 beta called canakinumab, which is an antibody that blocks it. It's also particularly interesting because Novartis funded a large 10,000-person randomized controlled clinical trial of canakinumab to prevent myocardial infarction in individuals who are at a high risk, who've already had a prior MI and have evidence of an inflammatory state on the basis of high levels of C-reactive protein. That trial met its endpoint and decreased the rate of cardiovascular events, and was published in the New England Journal in 2017. They didn't have any idea, of course, at the time that clonal hematopoiesis could have anything to do with this, but after seeing our study, they have actually gone back and sequenced 4,000 cases from their study. They haven't published that yet, but they presented it publicly at the American Heart Association meeting recently. And the individuals with TET2 and DNMT3A mutations had a major benefit from this drug, about a 60% decrease in the rate of cardiovascular events. Those without those mutations had a minimal effect on the decreased rate of cardiovascular events. So I think a nice independent validation of the role of these mutations in cardiovascular disease, but also a tantalizing hint that we might be able to intervene therapeutically and might be able to identify those individuals who would benefit from targeting that therapy. For a variety of complicated business reasons, that particular drug is not going forward to FDA approval, but there are new drugs targeting either interleukin-1 beta or upstream in that pathway targeting the NLRP3 inflammasome for the treatment of cardiovascular disease, hopefully targeting these individuals with CHIP. Okay, my title was about evolution of MDS, so I'm going to just talk about one of the avenues, which is really more related to therapy-related MDS in individuals with CHIP. So we in a number of groups were curious whether having CHIP could predict the development of therapy-related MDS. We initially looked at this in individuals with lymphoma who are getting autologous stem cell transplants. Those individuals have a very high rate of therapy-related MDS and AML. And so we sequenced patients right before their autologous stem cell transplant and looked to see whether CHIP predicts the development of therapy-related MDS and AML. And it did quite powerfully. There was a striking increased risk of therapy-related MDS and AML in those with CHIP compared to those without, and 14% of those with CHIP developed therapy-related MDS or AML within 10 years, which is a pretty high number for such a lethal, poor prognosis disease. And that translated as well to a major difference in overall survival. Some of that overall survival difference was due to therapy-related MDS and AML, but it was statistically significant difference as well in the rate of cardiovascular disease in those individuals with CHIP, indicating that simply knowing about that might alter other aspects of follow-up care. And I should add that Andy Bruner at MGH and others have looked at cardiovascular disease in MDS patients and find a significant increased risk of cardiovascular disease in MDS as well as in CHIP, so may start to think about other disease states. We think that this will not be limited to just cardiovascular disease, that may be that MDS patients may be particularly at risk for in addition to individuals with CHIP. So what are the mutations that are particularly relevant in therapy-related MDS? This is from work from Coleman Lindsley when he was in my lab, looking at a large cohort of individuals who underwent allogeneic stem cell transplant for MDS. And the mutations most associated with therapy-related MDS were TP53 and PPM1D. PPM1D is a phosphatase that regulates P53 as well as a number of other components of the DNA damage response pathway. So this gets to a model that Dan Link and Terrence Wong and others proposed in an elegant paper in Nature a few years ago, that while we used to think about therapy-related MDS and AML as the mutagenic process of chemotherapy or radiation introducing a ton of mutations in cells and that being the primary driver of the disease, that another model is possible where there is a pre-existing clone that we now think of as CHIP with a, say, P53 or PPM1D mutation. Those cells are exposed to an enormous selective pressure of myelosuppressive chemotherapy or radiation that selects for that mutant clone. That clone gets bigger, allowing a greater number of cells available for acquisition of additional mutations, such as a biallelic P53 mutation or other hits, leading to ultimately to therapy-related disease. So I'm going to finish with a little bit more about P53 in general. We've been interested both in PPM1D and P53, but we were kind of drawn to P53, though a little intimidated by the field for a number of reasons. One is P53 is the most commonly mutated gene in cancer. It's also the most studied gene in the human genome. There are 95,000 papers on P53 in PubMed. It plays, if there's one gene is responsible for the most morbidity and mortality from cancer, it would be P53. It plays a central role in genome stability, but it's a checkpoint for many processes. And it does not have a random spectrum of mutations. There are particular hotspot mutations, and the relevance of that actually has remained a major debate, and that's where we decided to weigh in. In the case of MDS, it is associated with a complex karyotype, high blast count, thrombocytopenia, resistance to many of our therapies, including lenalidomide, radiation, and chemotherapy. It's associated with a striking response to hypomethylating agents, though those patients will eventually recur with their disease. And it's associated with a horrible survival, worse than any other gene mutation. And so from the very first efforts that Raphael Behar made when he was a postdoc, that gene stuck out as the worst of the worst. It's like the most evil gene in the genome with respect to MDS and cancer in general. Here's the data more recently from Coleman's study in the setting of allogeneic stem cell transplant. By far the biggest predictor. We did a recursive partitioning model, and the first thing that split survival by far was P53, no matter how we looked at the data. So P53 is an extraordinarily powerful predictor of outcome. There's one silver lining in this slide, which is that not all individuals with P53 mutation die after their stem cell transplant. About 20% have long-term survival. Maybe those are the individuals that if we control their disease long enough to have a graft versus MDS defect, that they would do very well. Maybe there's something special about those P53 mutations that are different from others. We didn't see any in this initial, but that still remains possible. But that I think offers some hope that whether the use of hypomethylating agents or other manner to keep that clone in check long enough to have an immunologic benefit from the transplant provides some hope that this may be a treatable disease. So the mutational spectrum is not random. This is from cancer in general. This is from MDS in particular. And you can see that these mutations here all occurring, these are missense mutations, occurring in the DNA binding domain at particular residues, at particular missense mutations. The exact same residues are hotspots in MDS as well. Frame shift mutations or nonsense mutations that cause loss of function of the gene are scattered throughout, but the missense mutations always occur in the DNA binding domain, and particularly specific amino acid changes. So is that telling us something special about the biology of P53, the biology of those individual mutations that would provide some clues? So the approach that we took is this is just as CRISPR genome editing, in particular HDR, homologous recombination directed repair, was becoming possible. So we could systematically introduce all of those missense mutations in an isogenic system so that we could compare them and understand whether those mutations are leading to a gain of function, which is the subject of at least a dozen Nature papers, though all with different mechanisms, or was a dominant negative function. And we played a couple tricks there. K562 cells are actually P53 missense mutant, and we could back mutate it to wild type, have a fully functional P53 system, and then reintroduce these mutations, or excuse me, 13 cells which are diploid for P53, and we could introduce either heterozygous or homozygous mutations into that system. And then we did a wide variety of studies here, most of which I won't have time to talk about, but I'll just show you a little bit. So first we looked at DNA binding activity, and you can see that when we pull down P53 in wild-type cells, either treated with daunorubicin or with DMSO, we get a set of genes where P53 sits on their promoter, and those are the canonical P53 target genes that have been studied for decades. However, with these mutations, for the most part, there is essentially no DNA binding activity at any promoters. There are some mutations that retain some, and one mutation actually that has even more peaks. But when you do RNA-seq, those genes where P53 binds with this mutant are not expressed. So that is not an organized, guided DNA binding activity of mutant P53. That is an altered DNA binding specificity caused by the amino acid change that does not lead to any functional consequences that we can see. When we use gene expression profiling, by far, the major consequence is a loss of function, not a gain of function, as has sometimes been hypothesized. So these are the canonical P53 target genes. Either complete knockout of P53 or all the missense mutations lose those transcriptional targets of P53. I should note that P53 is a transcription factor, and that there is no consistent pattern of genes that are activated by all of the different mutations. They all activate a slightly different set of genes, nothing consistent. And that this is actually a dominant negative effect. So when we introduce, so here are the peaks for where P53 binds in a wild-type cell. If you knock out only one allele of P53, you still get DNA binding of the intact allele. So there's still P53 activity in a heterozygous loss-of-function cell. In a P53 null cell, of course, there's no DNA binding activity. But if there's a heterozygous missense mutation, you lose almost all the DNA binding activity, consistent with a dominant negative function. And if you have a missense crossed to a null allele, so there's no wild type at all, then you don't get anything. So those residual peaks are probably from the residual wild type there. And that translates into P21 activation. Then we did a very large set of in vivo studies with all of the combinations of genotypes that we see in MDS patients. We see wild type, we see heterozygous knockout of P53, homozygous knockout of P53, a missense mutation with an intact wild type allele, a missense mutation with a null allele, like deletion of 17p on the other allele, and with two different missense mutations. And we competed all of those to understand the hierarchy, and we gave that a selective pressure by doing this as a competitive transplant and then doing sublethal irradiation to provide a selective pressure for the mutant P53 and then following them over time. So I'm going to go, this is the last data slide, but I'll walk through this quickly. So here is the null cells compared to wild type, and the null cells outcompete very nicely. Heterozygous versus wild type also compete very nicely, and homozygous knockout compared to heterozygous knockout, homozygous still has an advantage. So many MDS patients lose both alleles of P53. That must mean that there's a selective pressure to losing the second allele if one allele is already gone. Here is heterozygous knock-in of one mutation, of a missense mutation compared to the wild type, and there's a competitive advantage. Heterozygous knock-in compared to a heterozygous knockout, and the heterozygous knock-in, the missense mutation wins, consistent with the dominant negative again. But a heterozygous knock-in crossed with a null allele compared to null, there's no difference. So if there was a gain of function, that's where you would see it, because you'd see a gain of function in addition to the null. So that basically shows a gradual continuum of the spectrum of P53 mutations from wild type to P53 heterozygous loss, P53 missense mutation crossed to heterozygous, and then ultimately missense crossed to a null, or null being identical. So those were the points that I was hoping to tell you a bit about today, that CHIP is associated with increased overall mortality, increased development of hematologic malignancy, increased risk of cardiovascular disease. We've also studied thrombosis in the setting of JAK2 mutations, and increased risk of therapy-related MDS and AML. And in the setting of P53 mutant MDS, this is the worst of the worst in terms of the molecular phenotype. They have extremely poor prognosis, complex karyotype, resistance to therapy. It's common in therapy-related MDS. And the missense mutations in the DNA binding domain, which are the most common types of mutations that we see in MDS, cause a dominant negative effect without evidence of a gain of function. And wanted to highlight a number of individuals. Sid Jaiswal led the work on CHIP. Stefan Bottcher, and Sid is now an assistant professor at Stanford. Stefan Bottcher led that P53 work I talked about and is soon to be running a lab in Switzerland, in Zurich. Susanna, who just was awarded the Bastianello Prize, and Coleman Lindsley, who is also running a lab now at the Dana-Farber. And I'm happy to take any questions.
So very interesting as always. So my question is about the TP3 mutation study. So you know, initially long years ago, people generated TP3 knockout mouse, which shows a very highly increased rehomogenation, but no substantial increase in epithelial tumors. But when the mutant allele is introduced in the TP3 null background, so many epithelial tumors. This explained by the dominant negative effect of TP3 mutant not to the TP3 biocell, but to homologous TP63 or TP73.
Yes.
But in your data, clearly shows, at least in mild compartment, this dominant negative effect is more confined to wild type TP53. So, but the mutation is very similar.
Yes.
So I'm wondering whether this
the same mutations. So that's the central question, and one we were very worried about from our reviewers. So I guess there's a couple answers. One is that those studies were generally done with overexpression of the mutant allele. We made endogenous edits, and I think those are different. There is forced interaction with other proteins, including those you listed, and other transcription factors that happen with forced overexpression that don't happen in this setting. We looked very hard for that, couldn't find it. So it may be an artifact of that. And then the second possibility remains that there is a gain of function in the development of epithelial tumors that's not present in our myeloid malignancies, which we've commented on, but we haven't looked, obviously, at epithelial malignancies.
Thank you.
Any other questions? David?
Thanks, Ben. Great talk. How do you explain the high frequency of lymphoid malignancies in your Jaiswal cohort? Because I think, unless you look at the supplement, you get the impression that this is all MDS and AML, but in fact, like 60% are lymphoid malignancies.
So first of all, small numbers there, and there are subsequent papers that will refine those risks better. The particular cohorts that we had in that initial paper did not have really well adjudicated myeloid malignancy or hematologic malignancy data. If you look at the actual diagnoses there, some of them were like cancer of the spleen and things that were clearly not fully annotated. But I think we'll get more refined risks. The two recent papers on specifically looking at AML have refined at least the AML risk very nicely. But why might lymphoid malignancies be increased? I guess there are a couple possibilities, and they both remain possibilities at this stage. One is that there are CHIP mutations in some lymphoid malignancies, and there have been some elegant case reports studied that show that there can be a hematopoietic stem cell clone that simultaneously lead to a lymphoid and a myeloid malignancy, and so you're increasing the risk of lymphoid malignancy simply by being the first hit there, which is a very interesting way of thinking about lymphoid malignancies, because we think about them as cells that acquired mutations in a lymphoid lineage, but the initial mutation may have occurred in a stem cell with the subsequent mutations occurring in a germinal center B cell or other cell. So that's definitely one possibility. The other is that there may be mutations that either reflect the microenvironment that increases, or the exposure of that individual, the risk of that individual that increases the risk both for CHIP and for the lymphoid malignancy, or that the CHIP clone alters the microenvironment of lymphoid cells in a way that is oncogenic. We've worked with Irene Gobriel on a large-scale analysis of myeloma patients who underwent autologous stem cell transplant. CHIP was very common in that setting, but it was not clonally related to the myeloma clone, which makes sense because myeloma cells have quite different mutations. So it may depend on the type of lymphoid malignancy as well. So in that case, we think it might be something more along the microenvironment level, but more work to be done there. Tim.
Ben, in your in vivo, the competitive transplant experiments, have you had a chance to introduce any of the P53 targeted agents like APR-246?
Not yet, but that's next. We're doing that a little bit in vitro first. The one thing that is actually a little interesting is that in those systems, azacitidine and decitabine don't hit the mutant clone. If anything, they increase the amount of the P53 mutant clone. My hypothesis there, which is completely data-free, is that the P53 mutations are associated with a complex karyotype, that complex karyotype requires enormous methylation changes for gene dosage effects, and if you drop a bomb on methylation and wipe it out throughout the genome, that you selectively hit the complex karyotype cells, which are usually the ones with P53, but then there is some reorganization epigenetically that allows those cells ultimately to recur, but don't have any data for that.
I think we'll have time for one last question. Anyone? No? Thank you very much, Ben Ebert. Great talk.